Okay, in this video I'm going to walk you through the steps of meiosis, but I'm going to leave out independent assortment and crossing over. I'll show you that in a different video. Okay, so let's start with our parental cell. This is going to be a 2N cell, and the imaginary organism that I'm going to use for this demonstration has 3 chromosome types. Here's type 1, type 2, and type 3. And since it's 2N, it has a copy from its mother in red. And this organism also inherited a copy of each chromosome type from its father in blue. All right, so there's the next step we need to look at is replication. Let's make sure we've got some replicated chromosomes. Okay, so there's the maternal versions of each chromosome type replicated, and there's the paternal, the ones from the father, replicated. Okay, let's skip forward now to metaphase 1 of meiosis 1. So this is where things get a little bit different from mitosis. At metaphase 1, let's give us a metaphase plate. Usually during mitosis, the sister chromatids would line up across from each other, but that's not what's going to happen here. Instead, we'll put the maternal DNA on this side. So there are the 3 chromosomes inherited from mom. And here's chromosome type 1 from dad, chromosome type 2 from dad, and chromosome type 3 that came from dad, lined up across from each other at the metaphase plate. Whoops, a little too far out there. All right, now we're ready for anaphase. And again, this is different from mitosis because in mitosis during anaphase, we saw the sister chromatids split. That's not what we're going to see here. We're going to see a different unit split. And so before we do that, I'd like to define this unit right here as what we call a tetrad. It's when the 2 homologous chromosomes that are duplicated are connected together. So this is the chromosome type 1 tetrad, and this down here is the chromosome type 3 tetrad. Okay. So when the tetrads split, it looks something like this. And you know what, I'd actually like to redraw my cell a little bit here to make it clear that this cell is in the process of splitting. Into 2. Okay. So let's keep going one more step and see these cells actually split into 2. Just a little bit smaller, give myself some more room. All right. So we need to do a little math here to come to grips with what has just happened. All right, so this is chromosome type 1, this is 2, and this is 3. 1, 2, and 3. How many chromosomes, how many versions of chromosome type 1 do we have in each of these cells? Well, the answer is just 1, although this is made up of 2 sister chromatids. It's actually just 1 chromosome. Of type 1. Right, and we just have 1 chromosome of type 2 and 1 chromosome of type 3. That makes this a 1N haploid cell. And same story over here. So after this is the end of meiosis 1, and we have 2 haploid cells. So at the beginning of meiosis, we started with a 2N cell, and we were 2N, 2N, still 2N because this cell had not split all the way, but then we finally got here to the end of meiosis 1, and we end up with 2 haploid cells. Now let's do meiosis 2 for both of these cells, just to be complete. So what's going to happen is, and I'll just start moving down here. We'll look at both of them. We end up this time with the sister chromatids actually lining up across from each other at the metaphase plate in meiosis 2. And then we'll see them split. Same story over here. This cell is also splitting. And finish this off with them completely split. And again, we need to do some math, some chromosome math here to talk about whether we're 2N or 1N. It should be clear by now that we only have 1 chromosome of each chromosome type, 1, 2, and 3, right? And so because we only have 1 version of each chromosome type, that makes each one of these daughter cells 1N. Okay, and before I end this video, I want to just point out that these are the gametes, all right? So this is the process of making eggs if you're a male, or if you're a female, and sperm if you're a male. And this is, the way I've shown this is really unrealistic. because there's no variation, there's very little variation. There's only two types of gametes that this individual can produce. So on the left, you're looking at two gametes that contain all of the DNA this individual inherited from their mother, all that DNA that's in red. And on the right, you see that the two gametes that were produced contain strictly paternal DNA, the DNA that this individual inherited from their father. So let's look at how independent assortment and crossing over give us the pretty much infinite variation that actually exists in the gametes that are produced by an individual in a separate video. But before I do that, I want to go over the review questions. What is the ploidy of the parental cell at the start of meiosis? Okay, so let me shrink this again so we can see the whole thing. And so you can see at the beginning, we start with a 2N diploid cell. The next question is, how do chromosomes line up during metaphase, and what actually separates during anaphase in meiosis 1? And so that's what we were talking about here, where the tetrads line up at the metaphase plate. So it's you could also say that it's the homologs lining up across from each other at the metaphase plate. You could say that the tetrad splits during anaphase, or you could say that the homologous pair splits at anaphase. Either way is correct. Actually, you should remember both of those in case I ask it in either of those ways on an exam. What is a tetrad? And so I've circled the tetrad, but just to explain it, it's the two versions of a chromosome type, and both of them have to be duplicated, right? So you end up with a grand total of 4 chromatids all together. That makes a tetrad. So the 4 chromatids of chromosome type 1 is the type 1 tetrad. All right, the next question, at what point does the conversion from 2N to 1N occur during meiosis? And so at the point of cytokinesis during meiosis 1, we go from 2N, a 2N cell, and then once we've split into these two separate cells, they are 1N. So that's the cytokinesis at the very end of meiosis 1. And the last question here is, what is produced at the end of meiosis 2? So these are 4 daughter cells. That are all haploid. Or 1N.